So first of all, I'd like to, on behalf of the organizer, I'd like to express our warmest welcome to the speakers and all the audience for coming to Sendai and also attending the symposium named New Horizons in Food Science by Agricultural Immunology. So as Dr. Maeda introduced, now this symposium named New Horizons in Food Science by Agricultural Immunology is held in a department, Graduate School of Agricultural Science. And now this stage 2 is now starting. And as Dr. Makino mentioned, CFAI is now is contribute to the organizing this symposium, and Dr. Aso is the director of CFAI, and Dr. Kitazawa and I are vice directors of CFAI. CFAI is the abbreviation of International Education and Research Center for Food and Agricultural Immunology, and it was established in 2015. And probably you have this leaflet of CFAI in your bag, and I took this image from the leaflet, and here there is the goals of the activity of CFAI. And if I magnify this sentence, it says, establishing a food safety system based on agricultural immunology to produce healthy agricultural products, livestock, and marine products. So in this sentence, the term of agricultural immunology would be a quite noble term, but this doesn't mean, this doesn't just mean the acquired immunity, but this word, this new term, also contains various potential abilities of agricultural organisms, including, for example, innate immunity, response to pathogens and stresses. It contains, for example, signaling and gene expression, and also metabolic homeostasis and maintenance of genome integrity, and so on. So now CFAI tried to do such very broad range of researches. And to achieve this mission of CFAI, we have 4 divisions in our center. And first is agricultural immunology, and the second is safety and function evaluation. And the third is cooperation with society. The fourth is research and administration. CFAI also has an agreement with foreign universities, and at this moment, this foreign university is in USA, the Netherlands, China, and Argentina. Of course, we are now trying to increase the number of the foreign university to do collaboration. And in this stage 2, this division, safety and functional evaluation, contribute to the organization of this symposium mainly. So this symposium is named as Food Safety and Functional Evaluation. And I guess these 2 topics might be main topics to be discussed in this symposium. And at a glance, these 2 looks rather simple, but for example, if you want to discuss about efficient and safety-conscious food production, we need much information, for example, about characteristic of agricultural organisms, but it probably contains gene and susceptibility to pathogens, and also responses to stimuli, stresses, and pathogens. And also, we know how we can apply this information to the production of to the food production. And in the case of detection and evaluation of harmful and beneficial ingredient of food, we need information about molecular targets of these ingredients and also functions in cells and also effect on human health. Of course, effect of human health has negative and positive result, positive effect by this ingredient. So in other words, these kind of very various field of researches are relevant to food production and food evaluation. So therefore, in this symposium, we invited distinguished professors from various fields of science. So for example, today we will have these talks, and we have talks by Dr. Gasser, Dr. Yanagida, Dr. Wigeltis, Dr. Shima, Dr. Ito, and Dr. Yoshioka. Of course, these speakers are distinguished in various fields, and I'm very happy to have these talks today. And tomorrow we will have these talks by Dr. Turner, Dr. Kato, Dr. Van Eden, Dr. Ohinata, and Dr. Shirakawa. And after finishing this talk, the young program of this symposium will start. And tomorrow in the afternoon, we will have poster session and introductory talks. And in 9th of August, we have youth program lectures. And concerning these lectures, Dr. Nakagawa will make introduction about these talks in the morning of 9th of August. So we also have these social events. For example, today in the evening, we have welcome cocktail. And tomorrow at lunchtime, we have lunch gathering. And also in the evening, we have banquet. So all the participants are welcome to these social events. So for details, please ask to the reception of this symposium. So with the following several slides, I'd like to introduce one of my research subjects. And now I have planned to contribute the mission of CFAI through their research on epigenetics. Epigenetics is a regulation of genome functions by chromatin and the nuclear structure. As you know, DNA forms nucleosome structure in our cells. And the chromatin, the nucleosome is the basal structure of chromatin, and this chromatin is packed into the cell nucleus in a highly organized manner. So this structure is very important for the regulation of genome function. And especially this nucleosome structure is a fundamental structure of epigenetics. Nucleosome is formed by these 4 kinds of histones, H2A, H2B, H3, and H4, and DNA is wrapped around these core histones and forms this kind of structure. And the status and the structure of chromatin, this nucleosome, is very important to regulate the gene function, including gene expression, DNA repair, chromosome segregation, DNA replication, and so on. So this is one example of the regulation of gene expression by histone modification. And in this case, I show the acetylated histone by histone modification complexes. And if these histones have acetylation, the genes usually get activated. But on the other hand, some methylation makes gene inactive. In this slide, I show the enzymes which are involved in the acetylation of lysine residues of histones. And this acetylation, this histone acetyltransferase, abbreviated as HAT, acetylate lysine of histone, and HDAC, this is a histone deacetylase, deacetylate the lysine, acetylation on lysine residue. So now it is known that some food ingredient makes effect on the function of this HUT and HDAC. So for example, in this article, the authors mentioned that some food ingredient and also product in gut microbiome affects the enzyme histone deacetylase and histone acetylase transferase. And finally, this food ingredient and product of gut microbiome affect in cancer and other diseases. So in my laboratory, now we are interested in acetylation, modification of H2AZ. H2AZ is a variant of histone H2A, and H2AZ is positioned in this part in the nucleosome. And H2A is evolutionarily conserved histone variant, and it is known that H2AZ is required for expression, DNA repair, and also mutation on H2AZ and overexpression of H2AZ is relevant to cancer. So we are interested in this H2AZ forms this nucleosome. And to analyze the function of H2AZ in cells, we knock out this H2AZ gene with chicken DT40 cell. And we establish cells in which we can knock out H2AZ in a tetracycline-dependent manner. In this case, if we add tetracycline, H2AZ expression is completely shut down. Inconsistent with this shutting down of the gene, the cell cannot grow anymore. This shows that H2AZ is required for cell survival. And if we check the gene expression in this cell, many cells were downregulated. So this means that H2AZ is required for the activation of a set of genes. So then we are interested in acetylation of H2AZ in the function of acetylation of H2AZ in cells. So to achieve this purpose, we expressed acetylation deficient H2AZ mutant in this H2AZ knockout cells. So this is a mutant, and in this mutant, we replace these lysine residues into arginine. So in this case, this mutant cannot get acetylation anymore. So we expressed wild-type H2AZ or this mutant H2AZ in cells. So both of these two H2A genes can complement the defect of cell growth. So this means that in terms of cell growth, this mutant functions in cells. So we are interested in the involvement of this acetylation, H2AZ acetylation on gene expression. So we check the expression of genes, and I show the example of EGR1 gene expression. EGR1 is one of transcriptional regulator and also known as a tumor suppressor. And EGR1 gene is quite rapidly activated after stimulation by serum. So in this experiment, in wild-type cells, EGR1 expression was induced quite rapidly within 30 minutes after serum stimulation. But in H2AZ knockout cells, this stimulation is significantly impaired. This means that H2AZ is required for the response of genes, response of these genes against the stimuli, environmental stimuli. So then we got interested in the contribution of acetylation of H2AZ in this reaction of EGR1. And we expressed this gene, this mutant H2AZ in H2AZ knockout cells. So in this case, we can replace all the H2AZ molecules into this mutant. And in the case of wild type of H2AZ, this expression of this wild type H2AZ can complement the activation of can complement the defect of EGR1 gene expression in H2AZ knockout cells. But this acetylation deficient mutant cannot complement this defect. So this means that H2AZ acetylation is required for EGR1 expression in response to environmental stimuli. And since EGR1 is a transcription regulator and also a tumor suppressor, so this means that HTZ acetylation has roles on gene expression, gene regulation, and cancer suppression, etc. So as I mentioned, now we know that many of food ingredients affects the function of enzymes on histone acetylation. So this means that food ingredient affects this gene expression of the cancer suppression, probably partly through the H2AZ function. So now we are continuing this experiment, and in the near future, I want to apply this finding to the evaluation of food ingredient. So in the last part of my talk, I like to mention about this person. Probably all Japanese know about this person, and his name is Date Masamune. This is his first name. This is not date, but Date Masamune is the name of this person. And he is a former lord of Sendai. And this year is the 450th anniversary of his birth in Sendai. And he developed the base of Sendai city. And he built a castle near this campus, and it was named as Aobajo. And this Aobajo comes from the name of Aobayama. Aobayama is the name of this hill. And of course, Date Masamune watched the Sendai city from the castle, and also he watched the world at the time. I mean, 400 years ago, it's a long, long time ago, indeed, he made this kind of Western-style ship and sent this person, Hasekura Tsunenaga, to Rome, probably to get information about European culture and also technologies. And yes, it was successful, but it takes 8 years. It takes long. But finally, in Rome, this Hasekura Tsunenaga was presented to the Pope of the Rome, and he brought many important documents. So now these documents are registered as national treasure, and we can see it in the museum in Sendai. And his figure is still in the palace of Rome. And anyway, this historical fact indicates that Date Masamune has internationality and very high innovative spirit. So to succeed Date Masamune's spirits, this time we invited these speakers from around the world. And I hope that your trip takes less than 8 years. But I'm very excited with the speakers from wide variety of scientific fields. And I believe that this stage 2 of the symposium, food safety and functional evaluation, becomes a memorable opportunity to discuss about the future of food science and life science. Yes, thank you very much.